So we'll take some questions. Maybe I'll start it off with kind of a general question that relates to NIEHS's interest in these the events that we're talking about today. So we heard about two model systems already, the zebrafish and the Daphnia, where there are that could potentially be used as a screening tool for agents that would induce copy number variation. That's a big interest to NIEHS because of the idea of Tox 21 using non-apical species for testing and so on. But we also heard some comments that there might be serious limitations on using these low organisms because of difference in genomic structure, organization, and function. Does anybody want to comment on that or have a little discussion on that?
So can I follow your question with a question for Scott?
Use your mic. Just push the button so that way everyone can hear.
So I'm going to answer your question with a question to Joe so that he'll respond to both of us, hopefully. So your gene duplications, exactly what is the mechanism driving those? Is it meiotic recombination, mitotic events? And the reason I ask that because it follows, and it's a correlate really of your question, unless we're looking at the same thing as an endpoint, it won't necessarily, or might not necessarily respond to the same environmental stressor.
So thanks for that question and putting it on Daphnia.
Well, you're close.
I'm close. I'll start by just one a little deflection in that we don't know for certain, but because of the life cycles of Daphnia, I think we can tease that out where we can revert from sexual reproduction to clonal reproduction. So I think that the answer is coming, and we don't yet know that answer. But to get back to your initial point of screening tools and what we can do with these, we're setting up large mutation accumulation panels with Daphnia where we're really can take 100 sisters and relieve them from selective pressures by removing offspring each generation. Passaging them forward, putting them through a bottleneck. And so after 100 generations, we can look through and begin to see that's about 10,000 genome events, exactly what these spontaneous de novo rates of CNV formation are. And we can do that in the presence of general stressors as well as specific stressors, and that's what we're doing.
Follow up when you're doing tests like HBRT or the cadmium, you're actually putting a biological selection on it. How much do you think that'll affect what you see and the mechanisms that are underlying it?
So for those mutation accumulation lines, we're doing them in organisms that have adapted to cadmium stress, where we know that there's not a selective disadvantage, there's not a fitness insult to those organisms, so that there's not that selective burden of cadmium. That stands as our control against our non-adapted populations where we hit them with cadmium, and we can begin to look at what's the effect of general stress, and we can tease out specific cadmium results from those adapted isolates.
So can I carry it a little further? When we were encouraged to wax and wane philosophical, and this is the time of day to do it. We'd be better with a glass of wine first. But so the term copy number variant is just a change in any amount of our genome. And we're using it for all sorts of things. And aneuploidy is a copy number change. But the mechanism that drives aneuploidy is very different from what Jim and I are talking about, for example. So there are different kinds of copy number variants. And I think that part of the answer to your question of what model organism can we use depends on what kind of copy number variant we want to look for. So gene duplication is important. As we look further and further in the human genome, we may find more of these very small, as Jim alluded to, exon changes. Single exon changes or gene duplications caused by maybe a different molecular mechanism than the hundreds of kilobase size CNVs that we're talking about that are associated with genetic disease. So to summarize all that, we have to kind of define, I think, a little better what we want their use to be for what kind of copy number variant. Make sense?
I guess I'm thinking about if a certain organism is good at responding in one way, it may not model what's going on in the human. And that
Exactly.
might be a problem for screening. Any comments on zebrafish?
I think right now, the work is still really ongoing as we work to try and define and get a better understanding of how those copy number variants are being formed in the fish before we can move forward with that, I guess, answering that question too. And each of our models, I think we have strengths and also weaknesses. And as we move forward and begin to have a better understanding of what's going on within the genome, then I think we'll be able to better address those questions and getting those to specifics.
I think what I'm hearing is that looking at just at the endpoint is not enough. If you're going to use it as a screening tool, is that fair or some assessment?
You're just going to.
Do you mean you want to look at the biological significance?
Well, if you just look at the readout, it's not going to really tell you what the mechanism is, right? And if you're using the readout as a screening tool, there might be some danger in that because you're going to miss mechanisms.
Correct.
Okay, let's open it up to Mike, please.
Not to pick on Joe, but there could be patient manipulations that why would that, what would we learn from patient manipulations in terms of that might give us I'm sure you
So the one answer to that is that we're doing this in the presence and absence of stressors, so that we can begin to look at specifically what are those impacts on mutation. And the other is that we're doing these specifically to look not just at SNPs, but to look at copy number variation, which there's not too many of these panels that have been set up for that purpose.
So are you going to exploit the sexual asexual differences there?
Oh yeah, that's absolutely what gives us the advantage to do these long-term.
Thank you.
Joyce Suji, Exponent. I think although this workshop is entitled Exploring Human Genome Plasticity, I think we have to keep in mind that these models are also useful for looking at effects on environmental creatures, and that you can't just generalize everything to humans. But there are some advantages even with these, in that it strikes me that a lot of what's being looked at right now are maybe high-dose effects on copy number. And I guess the next step would be, is there a dose response to that? And the advantage of using something like a Daphne or zebrafish is you could have, thousands and thousands of these organisms, and you can look at statistically whether you're getting an increase and whether there's any pattern with dose or multiple stressors or all these different variations, which are very hard to do with mice or humans. Is there any type of research to look at, can you go from this copy number variation to looking at dose response?
So I think you're absolutely right on. I mean, it could be, different model organisms or systems will be more useful than others for things like dose response. But I do want to make the point that, for example, it's relative what you call high dose, but for example, the hydroxyurea concentrations that we use to induce a lot of CNVs, we don't consider high at all. They're very, very mild. These cells have to keep replicating. Normally, in the case of people who are taking that same dose of the drug, and in cell culture, we get some decrease in survival, but bear in mind, I wouldn't call it severe.
Well, they're a therapeutic dose, and that's pretty high compared to what people normally get from environmental chemicals.
Yes,
but then there are a lot of environmental chemicals,
how you define it. Yeah.
And that's where it gets complicated. And I guess I had another.
Can I also make a comment
I'm sorry,
before we
go
move
ahead.
on real quick? Yeah, thank you. I also have to throw the question out there of what we're going to consider a dose response when we're looking at spontaneous, or when we're looking at copy number variation, because we may have, are we just going to look at the number of copy number variants that we're getting produced from these chemical exposures? Is that how we're going to relate dose response? Because we could just have a few copy number variants that appear but could have a larger biological significance than if we had, a few larger numbers that occurred.
That's true, and you wouldn't be able to detect it though unless you had a lot of organisms. And so that's the beauty of having species where you can use a lot of them. that you couldn't even do that with mice, what you could do with zebrafish or Daphnia. I had another question that might be because I'm naive, but there's a lot of different types of breast cancer. Would there be any reason to think that you'd get different types of copy number patterns, additions or deletions, whatever, with the different types of breast cancers? Estrogen positive, estrogen negative. Has anyone looked at that?
So we have begun to look at that using our data, and so we've segregated, and also some of our colleagues in Vancouver, British Columbia, Sam Avaricio and his group at the Genome BC Center. In fact, they just had a paper that was published in Nature a couple of months ago that looked at that in detail, and you can segregate breast cancer, or other types of cancer as well, but breast was the perfect example for this, into different molecular subtypes associated with unique patterns of gene amplification. And our own data would support that as well. Not only the different subtypes, but also the stage of progression. You see progressive accumulation of more and more chromosomal abnormalities.
Joe Gray has published on this, Anna Lisa Borenson-Dale, and those beautiful graphs that Randy showed that documented the amplifications and deletions above and below the line, they have those for the different subtypes. So it's very approachable.
So can I follow up on that question? I mean, it's not a question, it's a comment. So for breast cancer, there were many attempts to classify them into different subtypes, from like histological to pathological characteristics to gene expressions characteristic, and now to gene copy numbers variations and things like that. So has anybody used a system biology approach and an integrative approach, I would say, to overlay all these layers and find out, do they overlap, or do they actually, when you really truly boil it down to, then you can actually go back to just look at histology, like grays and things like this. I mean, or one or two driver or hubs, like for example, alpha positive, negative, triple negative. I mean, I know you have a lot of subtypes, but what would these information give? And do they align? Actually, the real question I want to ask is that whether based on those subtypes, we can kind of go backward and say this particular subtype of cancer was due to a particular exposure at a particular stage of life.
I'm shaking my head. I don't know of any studies that answer that very interesting question.
Comment question from somebody who's watching on the web, Dr. Alameda from University of Colorado. And he wants to comment on the suitability of model organisms, and he says that he's currently using yeast as a high-throughput model for screening. And his comment is that if you use it for somatic CNVs, that the outcomes are somewhat different from humans, and they're at lower frequencies, but for the meiotic events, they're really indistinguishable. So does anybody want to comment on that? You agree?
I have talked to Lucas about this, and I agree, and I know the data, and I think that's just wonderful, and it makes sense, doesn't it? Because yeast live and die by homologous recombination, and that's what you're going to see is the mechanism involved, whether it be mitotic or meiotic. I mean, yeast are very different from human cells in terms of the choice of repair pathways they're used, and they choose homologous recombination almost exclusively. Whereas we have more of a balance between end joining other pathways here. And homologous recombination. So, makes sense.
One more question?
Okay, Lou Gross, University of Tennessee. So one of the other workshops that this committee sponsored was specifically on issues of dealing with mixtures and responses to them. And it seems to me as if you have a collection of techniques that may allow us to deal with multifactorial responses to mixtures. And that also ties in with a upcoming workshop on big data. And I'm wondering if you have thought about that, or if there is a standard methodology for dealing with multifactors in looking at responses to multiple stressors, or is that really not done yet?
Well, we haven't. It's with our. I mean, not that we haven't thought about the myriad of different possibilities that might give rise to replication stress in seed and base, but we're limited by the capabilities of our assay system right now, which is the direct one of mammalian cells. It's expensive and time-consuming, and so sorting out things
Tom,
one at a time.
Did you try hydroxylamine with fitacolin and see if there was just no?
I mean, we have a long list on the bulletin board of things we would love to look at one at a time, let alone combining them. And it's, yeah, so we're trying to develop more rapid assays, strictly in mammalian cells.
Okay, so I guess we'll end it there. I'd like to thank all the speakers for a really, really interesting and exciting session, very informative, and thank the audience and people on the web for participation. Thank you.